So, you have to extract RNA. Which method do you choose? Today, we compare the protocols for the classic, TriZOL, the fast silica column kit, and the often overlooked low pH method. And we will also touch on two precipitation methods to get better yield with isopropanol, or better purity with lithium chloride. Protocol 1, TriZOL, or Phenol-Chloroform. TriZOL is a commercial name for a solution of guanidinium thiocyanate and phenol to denature proteins and inhibit RNases. The protocol starts with the homogenization of your cell pellet, followed by the addition of chloroform to induce phase separation. After centrifugation step to promote phase separation, three distinct layers are formed. RNA stays in the upper aqueous phase, genomic DNA moves mainly to the white interphase, and proteins move to the pink lower phase. Now, you have to carefully collect the upper aqueous phase without touching or disturbing the DNA layer below. To do that, angle the tube and with an RNase-free tip, slowly draw up the colorless upper phase and transfer it to a fresh RNase-free tube. Once the aqueous phase is transferred, proceed to the RNA purification by isopropanol precipitation or silica column. Protocol 2, Silica Column Kits. It operates on the principle that nucleic acids selectively bind to a silica membrane in the presence of high concentrations of chiotropic salts and alcohol. The sample is homogenized in guanidinium thiocyanate, mixed with ethanol and applied to the silica column. The high concentration of chiotropic salts and alcohol alters the hydration shell around the RNA molecules, allowing them to bind to the positively charged silica matrix. The bound RNA is washed multiple times with buffers and then eluted. Genomic DNA can co-purify with RNA, so many commercial kits include a DNase 1 treatment to remove contaminating DNA. This method is fast and simple, but might yield less RNA compared to TriZOL, but generally provides higher purity. Protocol 3, the low pH method, US Patent 5973137. The third method is a low pH protocol. This often overlooked protocol relies on the principle that pH affect the stability and solubility of DNA, proteins, and RNA. At pH 4, RNA remains stable and soluble, while genomic DNA and proteins denature and lose solubility. The protein DNA precipitation reagent precipitate these contaminants, leaving intact RNA in the supernatant for recovery via centrifugation. You can finish with isopropanol or a silica column. In my experience, this method is superior because it is non-toxic, allows for boiling lysis of tough samples, is highly scalable, and buffers can easily made in-house, using ultrapure water. The process is rapid, yield samples virtually free of genomic DNA, and works perfectly as a cleanup step to rescue previously extracted contaminated RNA. Precipitation choice. Whichever method you choose, once your RNA is in the supernatant, you can use isopropanol or lithium chloride to recover it. For maximum yield, use isopropanol. For maximum purity, use lithium chloride. Lithium chloride precipitates almost exclusively RNA, leaving residual DNA and proteins in solution. However, since lithium can inhibit polymerases, perform one extra ethanol wash to ensure your RNA is ready for downstream applications. One last warning. Even trace amounts of phenol can interfere with concentration measurements and skew the 260 to 280 ratio, as phenol absorbs strongly near the 260 to 270 nanometer range. Furthermore, if you use the Agilent 2100 bioanalyzer, residual phenol can alter the electropherogram, leading to inaccurate RNA integrity number. To summarize, TriZOL is powerful, but it is toxic and not practical if you have a large number of samples. It also carries the risk of phenol contamination, which can ruin your QC. Silica columns offer speed and ease of use, but they are expensive and often come at the cost of total yield. The low pH method is easy, cheap, non-toxic, and does not interfere with your QC. Just keep in mind that you have to prepare the buffers in-house. I have used this low pH method extensively for both tissues and viruses, and I'm glad I could introduce it to you. I will leave all the links in the description below. If you found this video helpful, please subscribe and like. My cat and I will say.